Have you ever found yourself wondering how genes control the production of proteins? Welcome to the fascinating world of gene expression, the process by which the genetic code, the nucleotide sequence of a gene, is used to direct protein synthesis and produce the structures of the cell. This process is essential in determining the biological makeup of all living organisms. It's like a finely tuned symphony where each instrument, or in this case, gene, has its specific part to play at just the right time. The regulation of gene expression ensures that proteins, the workhorses of our cells, are generated in specific cells at the right time and in just the right amounts. This precise regulation is what makes us who we are, from the color of our eyes to the rhythm of our heartbeat. Now, let's delve deeper into how this process is regulated. First off, let's tackle transcriptional regulation. This is a fascinating stage in the process of gene expression, where genetic interactions and epigenetic effects come into play. To understand it better, imagine a complex dance. In this dance, the dancers are transcription factors, and the dance floor is the DNA. These transcription factors interact with specific DNA sites, just like how dancers interact with each other. Depending on the rhythm, the dancers may enhance or inhibit each other's movements. Similarly, transcription factors can enhance or inhibit the binding of RNA polymerase, the enzyme that transcribes DNA into RNA. But there's another layer to this intricate dance. Enter the epigenetic effects. These are modifications that change the accessibility of genes, but they do so without changing the DNA sequence itself. Imagine if the dance floor suddenly became slippery or sticky. The dancers' movements would change, not because they've learned new steps, but because the floor's conditions have changed. In the world of genes, these changes to the dance floor are like DNA methylation and histone alterations, two common types of epigenetic modifications. DNA methylation typically reduces gene expression, while histone alterations can either enhance or reduce it. So, transcriptional regulation is like a dance, with its rhythm set by transcription factors and the conditions of the dance floor determined by epigenetic modifications. Moving on, let's explore post-transcriptional regulation. Next up is post-transcriptional regulation. This phase of gene expression regulation is all about making sure our mRNA is ready for the spotlight. You see, after a gene is transcribed into mRNA, it doesn't immediately become a protein. There's still work to be done. That's where post-transcriptional regulation comes in. It's like the final fitting before a fashion show, ensuring that the mRNA is in perfect shape and ready to strut its stuff on the catwalk of protein synthesis. One important aspect of this process is the modification of mRNA ends. These modifications act like bodyguards, protecting the mRNA from degradation. It's a rough world out there in the cellular environment, and these modifications ensure that our mRNA makes it to the protein stage intact. Then we have splicing. Think of splicing as the tailor of the mRNA world. It snips away the non-coding segments known as introns and stitches together the protein coding regions called exons. This process ensures that the mRNA is streamlined and primed for efficient protein synthesis. So post-transcriptional regulation is all about refining and protecting our mRNA, shaping it into the perfect template for protein production. It's a crucial step in the gene expression journey, and without it, our cells wouldn't be able to function properly. Now that we understand post-transcriptional regulation, let's discuss translational regulation. Now, let's turn our attention to translational regulation. This stage of gene expression is where the rubber hits the road, where the genetic code is translated into proteins. But, just like a finely tuned orchestra, every instrument, or in this case, molecule, has a part to play, and the timing needs to be perfect. One of the key players in this process is a group of molecules known as microRNAs. These tiny regulators attach to specific sequences of mRNA. This can block the initiation of translation, essentially putting a pause on protein production. Alternatively, These microRNAs can cause the degradation of mRNA, like a molecular shredder, ensuring that no protein can be made from this template. But microRNAs aren't the only maestros in this biological symphony. There are also molecules known as translational repressors. These can bind to the RNA and interfere with the initiation of translation, effectively silencing the gene's expression. And then there are the various other factors that can influence the process of translation. These include RNA-binding proteins and specific sequence elements within the mRNA itself. These can act like a speed dial, controlling how fast or slow the protein-making machinery works. So, as you can see, translational regulation is a complex and highly coordinated process, ensuring that the right proteins are made at the right time. Lastly, let's look at post-translational regulation. Finally, we arrive at post-translational regulation. Picture this. Once the process of translation has occurred, we have a polypeptide chain. This, however, is not the end of the journey. These polypeptides need to be processed further to become functional proteins. This is where post-translational regulation comes into play. Post-translational regulation, much like a master sculptor, alters the structure and function of these proteins. How, you may ask? It does this by adding or removing chemical groups, a process akin to chiseling away at a block of marble to reveal a masterpiece within. Two such chemical groups are phosphorylation and acetylation. Phosphorylation involves the addition of a phosphate group to the protein. This can activate or deactivate the protein, effectively turning it on or off. Acetylation, on the other hand, involves the addition of an acetyl group. This process can alter the protein's interaction with DNA, influencing gene expression. These modifications, while minute, have a profound effect on the protein's activity and gene expression. They ensure that the right proteins are active at the right time, contributing to the intricate symphony of life that plays out in our cells every second of every day. So, there you have it, a step-by-step guide to the regulation of gene expression, a process that is as complex as it is fascinating.